This video, the aim of it is to give you an overview of what is recombinant DNA technology. So what I want you to really learn from this slide is the definition, what is recombinant DNA? Recombinant DNA essentially is when DNA is combined from two organisms, is called recombinant DNA. Now recombinant DNA technology, so this allows us to manipulate and transfer. But the definition is very important that you have to memorize, is recombinant DNA. What is it? DNA combined from two different organisms. When that recombinant DNA, when that is transferred to an organism, that organism becomes transgenic. Or we can say they become GMOs. GMO stands for genetically modified organisms. I want to start this topic with using the example of diabetes and insulin. Now we know diabetic patients, type 1 mainly, they require this insulin injection. And insulin is so important for diabetes. How do we make insulin? So before, insulin was made using animal extractions, and that was quite challenging. It was timely and also cost a lot, risk of infection and immune rejection as well. So scientists, they thought we need to develop better ways, and this is what we have: recombinant DNA technology. I want you to understand the basic concept here. So we know that insulin is a protein, it's made from amino acids. Now what we need to do is we need to isolate the insulin gene from the human being. So this is a human cell, and then we have extracted this insulin gene, and that is called isolation. Now at the same time, we get a bacteria, and bacteria, we get that plasmid out of bacteria, and then what we do is that we isolate the plasmid, and then we add the insulin gene into the plasmid. Here we go. Now this is called recombinant plasmid, and we've covered that. It's got two different genes from two different organisms. Now this recombinant plasmid, which contains insulin gene, is now transferred into this bacteria again. And now the bacteria is called transgenic bacteria because it has this recombinant plasmid, and that is called transformation. So we've done isolation, done insertion. Insertion was the vector here, this is our plasmid vector, and then we've done transformation. Number 4, we're going to skip it here for now, but later on we'll go over it, identification. And number 5 is these bacteria, they grow and they multiply, and at the same time as they're multiplying, they are making insulin. Why are they making insulin? Because they have insulin gene. And bacteria can make proteins because they contain ribosomes. They do transcription, translation. Why do we choose bacteria? Because they multiply very fast. Exam question. Give 2 reasons why bacteria are able to use human DNA to produce human proteins. Our example is, how is it possible for bacteria to make protein from our insulin gene, from a human gene? How is it possible bacteria makes protein? Which protein? Insulin hormone. Number 1, you can say the DNA code is universal. So we have the same coding for the same amino acid in all species. And the mechanism of transcription and translation is universal as well, is same in bacteria. And we know that there's also translation occurring in bacteria because bacteria also have ribosomes as well. The next 3 videos will be going over isolation. So we see that this insulin gene, how is it really extracted from the human cell? How is this insulin gene extracted from all the other genes? And how is it mixed in with the vector? The plasmid is our vector. How is it mixed in? How does it really join to form this recombinant plasmid?